Hello, everyone. My name is Leilani, and today we're going to be diving into one of biology's most powerful electrical events, action potential. It's the body's language of electricity, allowing neurons to communicate, muscles to contract, and thoughts to form. Understanding how ions create these electrical spikes connects directly to medicine, anesthesia, and neurophysiology education. By the end of this presentation, you should be able to 1. Explain how ionic and electrical changes produce an action potential. 2. Describe how these changes allow neurons to send messages. And 3. Apply this understanding to real clinical and teaching examples. According to Sherwood 2015, mastering this flow from resting to active states is the foundation of all nervous system physiology. The resting membrane potential. Inside of the cell is about negative 70 MV compared to the outside. It's maintained by the Na and K plus ATPase, which is 3 Na plus out and 2 K plus in. The leak channels are negatively charged and proteins maintain the potential. At rest, a neuron is like a charged battery. Inside is going to be that negative 70 millivolts relative to the outside. Now, in order for the ATP pumps to use the energy to move 3 sodium ions out for every 2 potassium ions brought in, is based off of Sherwood in 2015. Because potassium leaks out more easily and negatively charged proteins remain inside, the cell stays negative. You could think of it as cocking a spring. Energy is stored and ready to fire, but it will not act unless it is stimulated. Now, depolarization. It is also known as the initial stage of the action potential. Now, a stimulus causes the voltage-gated Na plus channels to open, a rapid increase of Na plus entering the cell. The membrane potential increases to approximately plus 30 MV. And then our all or none law, which is once the threshold is approximately negative 55 MV is met, it must occur. Now, according to Sherwood of 2015, like I said, when the stimulus pushes the membrane to the threshold, the voltage-gated sodium channels snap open. Sodium rushes in and the voltage spikes to around plus 30 MV. This is an all or none event. It either fires completely or not at all. I like to compare it to a light switch. You can't get half a light on or a dim light unless you flip it on. The whole circuit must activate. Which brings us into repolarization and hyperpolarization. Now, immediately after the spike, those sodium channels close and the voltage-gated potassium channels open up. Sherwood 2015. Potassium flows out, bringing the voltage back down. That's repolarization. Sometimes it overshoots below negative 70 MV, which will create a hyperpolarization. During this phase, the neuron enters its refractory period. The absolute refractory period ensures a signal moves only one way, while the relative period sets a limit on how often a neuron can fire. Propagation of action potential. Now, the excitation travels. When one segment depolarizes, it triggers the next section to open its sodium channels. In myelinated axons, the impulse jumps between nodes of the Ranvier in a process called saltatory conduction, per Sherwood 2015. That jumping can reach speeds up to about 120 meters per second. Diseases like multiple sclerosis damage myelin and show how structural changes can slow or block signals entirely. Integration at the synapses. So when the action potential reaches the axon terminal, voltage-gated calcium channels open up and Ca2 plus rushes in via Sherwood 2015. Calcium causes vesicles to fuse with the membranes and release neurotransmitters into the synaptic cleft. Those chemicals bind to receptors on the next neuron, creating either an excitatory EPSP or an inhibitory IPSP effect. The axon hill lock sums thousands of these inputs to decide if another spike fires. It's how thought and movement emerge from our microscopic yes or no signals. Now, let's apply this to lidocaine. Now, lidocaine blocks voltage-gated sodium channels so that depolarization can't occur via Sherwood 2015. Without an action potential, pain signals never reach the brain. That's why dentists use lidocaine to numb tissue locally. The sodium channels stay closed and no electrical wave forms. It's a beautiful real-world illustration of how physiology becomes pharmacology. Here we're going to go into our scholarly source. This is our introduction. Now, in this review of the article by Fowler in 2022, the author discusses the use of active learning as an educational strategy for teaching action potentials and their clinical application in neuroscience medical education. The purpose is to use active learning to instruct students on action potentials. The challenge is students cannot comprehend the visualization of ion movement. And lastly, our interactive classroom model solution is what we're going to be going over. So in order to reinforce these concepts with research, now, the medical students frequently memorize the order of the ionic events without being able to understand them. Fowler 2022 created an interactive classroom activity lasting 10 minutes. Students were divided into small groups and acted out the role of neurons and ions, simulating changes in voltage. The method. There are 52 students enrolled in a neuroscience course, and they all participated in the study. Each group of students engaged in the human neuron simulation. As students moved to represent Na plus and K plus fluxes through the membrane, they also represented the opening and closing of the membrane gates via Fowler of 2022. So the students were given the roles by the instructors. Some of them were Na plus, others were K plus, and the membrane sections. So when the Na plus students entered, other students exited, demonstrating depolarization and repolarization. Observers documented the correct sequence of students based off of their movement that was shown by Fowler in 2022. Here are some of our results for figures 1A and B, modeling both the depolarization and repolarization. So in panels A and B, the students act out the membrane events. Once the Na plus ion steps into the membrane becomes depolarized. When K plus ions leaves, the membrane becomes repolarized. Fowler of 2022 state that this movement sequence allows students to correlate ionic flow with voltage changes. Now here is figure C. As the Na plus and the K plus pump phase resets membranes, sequential activation models signal propagation. So here you can see in panel C, it depicts students returning to their original position, which represents the Na plus and the K plus pump. As the instructor proceeds down the line of students, the wave is carried similarly to a real axon. Following this transition to a discussion of the clinical correlates of multiple sclerosis and neurotoxins via Fowler of 2022. Now, the integration and summary. The physiological explanation of how electrical mechanisms occur, the physiological explanation of how to teach electrical mechanisms effectively, and the pedagogical explanation of how to integrate science into lasting student understanding. Now, this is active learning, improved concept mastery, and engagement. The students relate action potential to clinical topics, and it's recommended for medical and nursing curriculums. Fowler of 2022 stated that students were able to retain the material better and articulate the neurophysiological concepts using their own words. Active learning created a connection between the theoretical aspects of teaching and practical application of knowledge. Sherwood of 2015 describes what occurs electrically. Fowler of 2022 describes how to teach it effectively. Science and education combined create deeper understanding. These two references tie together both physiological knowledge and the pedagogical techniques. Sherwood in 2015 describes the mechanisms involved, while Fowler of 2022 illustrates how to apply those mechanisms to an educational environment and bring them to life for the learner. Which brings us to our conclusion. The action potential flows a standard sequence: rest, depolarizes, repolarize, hyperpolarize, and rest again. Myelination and refractory periods increase the rate and direction of signal transmission. Regulation of ions underlies all neural communication. And lastly, disturbances rest in disease or therapeutic targeting. Now, I know that was a lot of information that we just went over. So just so that we can have a basic recap, here we go. I want you to think of your brain and all your nerves as a small network of very fine electrical wiring, allowing your body parts to communicate with one another. So this is how it works. We have our nerve cells, neurons, and they are essentially batteries. They have one side slightly negative and the other side slightly positive. Then we have an event that occurs. Let's say you touch a hot pot. An impulse begins to flow through the circuit and flip the switch on that battery. The nerve then opens its ion channel gates, which allows sodium and potassium ions to rush in and out. The rapid exchange of these ions creates a brief burst of electricity, which becomes a wave or a domino effect that travels along the length of the nerve. Now, this wave is called an action potential, and this is how your body sends messages quickly. Therefore, when you touch that hot pot, the wave will travel from the fingertips to your brain, and the brain will send a similar wave back to your finger, telling your hand, hey, that's hot, don't touch it. So in summary, action potentials are your body's tiny lightning bolts. They are extremely rapid electrical signals used by your brain and the rest of your body to communicate with each other. Here we have my reference list. Through this presentation, I have explained how neurons use electricity to communicate and how modern teaching methods make these concepts visible. Through the first half, reviews of the physiology of the action potentials were via Sherwood of 2015. And in the second half, we went ahead and analyzed Fowler's of 2022, which was a scholarly study on teaching this concept through active learning. Both of these two sources demonstrated both scientific accuracy and innovative teaching, which meets the school's criteria for academic and scholarly integration. Well, that about wraps it up for me. If you have any questions, comments, or concerns, please feel free to reach out. And I hope you enjoy the rest of your day. Have a good one. Bye-bye.